Hello, everyone. Welcome to our Chapter 2 lecture. Today, we are going to explore some of the highlights from Chapter 2, which is on the topic of biological psychology. Go ahead and get that pulled up here. So Chapter 2 deals with the topic of biological psychology, but more specifically, the structure and function of the central nervous system, particularly the brain. So we're going to look at some of the foundational concepts here in this chapter that I think are most important for you guys to be aware of in an intro-level course. So we'll start out by describing what the nervous system is, what it does, and how it's laid out. So your nervous system is, of course, the network of billions of neurons. Those are the cells that comprise the basic unit of the nervous system. There are other types of cells that support neurons, but neurons are the stars of the show, so to speak, when it comes to the central nervous system. So neurons are the basic structure that comprises the nervous system. You have around varying, the amount of neurons that a person has varies depending upon their body weight, lifestyle factors. There are a number of things that go into the number of neurons a person has in their body, but I would say around 120 to 150 billion neurons in the typical human central nervous system. Of course, the bulk of those neurons being located in the brain, but throughout the body as well. So this network of neurons, the nervous system, is responsible for every thought you ever have, every emotion you ever experience, everything that you hold dear in life, everything about you as a person, at least according to the materialist viewpoint that most of modern psychology and neuroscience relies upon. All of those things that make you you and determine what you do on a daily basis are essentially determined by the interaction between your brain and the world around you, which your brain is responding to. So it's really a mind-boggling thought to think about that all of these things that we hold dear about ourselves that make us people are essentially the result of the electrochemical activity of several hundred billion neurons. So that is the assumption that we're working from in this course, the materialist assumption that we talked about in Chapter 1. All right, I was having some trouble getting switched over to the next slide there. So here we see the layout of the nervous system, the way that it's broken down into its various subsystems. So most of what we're going to talk about in this chapter and throughout the course, with seldom exceptions, will relate to the brain, and that is part of what is called the central nervous system. So the central nervous system is comprised of the brain and the spinal cord. However, there's more to the nervous system than just the central nervous system. You have to be able to connect the brain and spinal cord to the rest of the body, which thankfully is accomplished through the peripheral nervous system, which itself is broken down into two subsystems. So we have the somatic nervous system, which is the part of the nervous system that's responsible for your sense of touch, your ability to send messages to your muscles, all of your sensations that you experience in terms of hot and cold. Soma means body. So your somatic nervous system allows you to control your muscles and have a sense of touch, tactile stimulation, etc. But the other half of the peripheral nervous system is the autonomic nervous system, which is the part of the nervous system that controls automatic processes, hence autonomic nervous system. And it is divided into two subsystems itself. So you have the sympathetic nervous system, which is often thought of as sort of the fight or flight system. And then you have the parasympathetic nervous system that operates in opposition really to the sympathetic nervous system and is responsible for us maintaining a sense of equilibrium and bringing us back to a state of calm when we become aroused via the sympathetic nervous system. So when you encounter a threat in the environment, it's your sympathetic nervous system that lights up in that fight or flight sort of fashion when you get that adrenaline release and blood is diverted away from your digestive system to your muscles, and all of these things that mobilize us for a fight or flight response associated with adrenaline or norepinephrine and hormones like cortisol, for example. All of these relate to the activity of the sympathetic nervous system. On the other hand, the parasympathetic nervous system, as I said, is kind of the opposite of the sympathetic nervous system, hence parasympathetic, and it brings us back to a state of calm and keeps us at a state of homeostasis biologically, or what you might call a state of balance. So if you have someone whose sympathetic nervous system is overly active, then you have someone who's going to have probably a very anxious disposition, as opposed to someone who's more in tune with their parasympathetic nervous system, would be someone who would have more of a relaxed sort of disposition. But as I said, the star of our show in this chapter is primarily the brain. So most of what we talk about in this chapter and the rest of the course will relate to the activity that takes place within the brain, which is about a 3.5 pound, on average, 3 to 3.5 pound mass of tissue, of course, that resides between your ears. And as I said, it's the brain that's responsible for every aspect of our experience, everything that makes us a human being, every feeling and thought that we ever have. Quite an astounding thing to think about when you think about it. So, as I said before, the neuron is the basic unit of the nervous system. And here we have an actual microscopic slide of a neuron highlighted here. And you can see the structure of the neuron. And as you look at this circular structure here towards the upper center, that is the cell body of the neuron, where the nucleus and organelles are located. Essentially, a neuron is an animal cell like any other animal cell, but there's something very special about the neuron and the fact that it transmits these electrochemical signals that create our experiences and decisions in life. They are electrochemical signals that are operative, we might say, within the neuron. So today we're going to look a little bit at how those electrochemical signals operate within the neuron, and then we'll also zoom out more broadly to the various parts of the brain and the functions that they serve. So let's take a look at this point at the neuron. So the next few slides are going to basically cover terminology. So we have several terms here that are important. It's important that you familiarize yourself with the meaning of these terms. I'm going to go ahead and skip ahead to our slide that gives us a visual representation of these structures. So you can read these slides more in depth on your own, but I want to show you guys a visual of where all of these various structures are located. All right, so the basic structure of the neuron you can see here in a schematic fashion. You see that the cell body is that circular structure there towards our left when we're looking at the image. And this is the part of the neuron that collects and integrates signals from other neurons. So other neurons communicate with each other. They don't actually touch each other. They communicate with each other at a site called the synapse, which is the gap between two neurons. But essentially, it's the cell body of the neuron that detects, or we might say really calculates the inputs that are coming in from other neurons. So these inputs come in via structures called dendrites that you can see there. And these dendrites are kind of like branches coming off of a tree. And these dendrites, if you zoom up on them really close, they have these receptor sites that receive incoming neurotransmitters from other neurons. And what's actually happening there is that you have hundreds, if not thousands, of dendrites on a particular neuron that are reaching out to its neighbors. And those neighbors are releasing substances called neurotransmitters, which bind to the receptor sites on those dendrites. So neurotransmitters can have either an excitatory or an inhibitory effect on the neuron that is receiving those inputs. So at any given dendrite, there's going to be neurotransmitters, which are chemicals that are traded between neurons that are sent out by the preceding neuron. And when those neurotransmitters bind to those receptor sites on the dendrites, they will have the effect of either stimulating the receiving neuron or inhibiting the receiving neuron from being stimulated. So at any given moment, you have all of these contradictory inputs coming in through the dendrites. You have excitatory inputs and you have inhibitory inputs that are all coming in simultaneously, probably hundreds of each, if not more, maybe thousands, depending on the neuron, because neurons tend to have a lot of neighbors, particularly the neurons that are located within the brain. So all these incoming inputs are being detected by the cell body of the neuron. And at any given moment, if that neuron receives sufficient stimulation from its neighbors, if there are enough excitatory inputs that are coming in through those dendrites, then there are certain ion channels, potassium and sodium channels, that open up in the cell body of the neuron and cause an electrical discharge to be created by the cell body. So you receive sufficient stimulation at any given point, and that cell body is going to fire an electrical impulse down that long, narrow part of the neuron called the axon. So that axon is that long, narrow part of the neuron, and the axon of any given neuron, they vary in length. So depending upon the type of neuron that we're talking about, an axon of a given neuron could be microscopic in length, or an axon could be several feet long if it's an axon that goes from the top of your spinal cord to the bottom of your body. So axons vary quite wildly in their length. And the longer the axon is, of course, the longer that electrical signal has to travel down that axon, the farther it has to go. And the farther an electrical signal has to travel along a conductor, the greater the probability that that signal is going to slow down and degrade over that distance. So thankfully, we have this insulation, this kind of insulation around our axons called myelin sheath, which is made of this fatty substance. We usually just call it myelin, but this myelin substance is grouped together in these clumps that you see here called sheaths of myelin sheath. And little gaps between the myelin sheath that you see there are referred to as nodes of Ranvier. But these myelin sheath play a critical role in insulating the axon and keeping the electrical signal from degrading as it travels along the axon, to keep the electrical signal renewed and strengthened. And in fact, the longer the axon is, the more critical myelin sheath becomes. In fact, there is a disease called multiple sclerosis, or MS, which is characterized by the destruction of the myelin sheath. And what happens in MS is your immune system mistakes your myelin sheath for a foreign invader and comes in and starts devouring your white blood cells, and other immune system cells start to come in and devour the myelin sheath. And this ultimately causes the axon to not be able to transmit that electrical signal as well. And over time, as the disease progresses, people with multiple sclerosis that's really far advanced, there may be some degree of paralysis involved because neurons are no longer able to convey those signals effectively down the axon. And it can be and often is fatal in a lot of cases where it's really severely far gone. A lot of folks live with MS for many, many years and manage it. For other folks, it ultimately becomes debilitating and ultimately fatal. So there's a lot of variability in folks who have MS, but for a lot of folks, unfortunately, it turns out to be a fatal long-term degenerative disease that takes, in many cases, years and years to fully unfold. Neurodegenerative diseases are quite terrible, and they're vicious, and if we can find cures for neurodegenerative diseases like MS, Alzheimer's, and Parkinson's disease, just to name a few, then we could reduce quite a bit of suffering in the world. And this is something that's personal to me because I've had a number of family members who have suffered from neurodegenerative disorders like Parkinson's disease and MS and Alzheimer's. So I, as many viewers probably feel the same way, I would love to see these diseases eradicated, but we have a long way to go before we fully understand the genetics and the environmental causes behind it, and eventually work towards curing these neurodegenerative diseases. Perhaps gene therapy will come in handy one day in that arena. Anyway, I kind of got off on a red herring there about MS. So you have the axon, which is transmitting the electrical signal down that structure to the other end of the neuron. And on the other end of the neuron, you have structures called terminal buttons. And these terminal buttons are actual little vesicles that contain neurotransmitter. These are the neurotransmitters that allow neurons to communicate with each other. So the electrical signal reaches the terminal buttons and causes those terminal buttons to release their payload of neurotransmitter into the synapse, which you see there. And when that neurotransmitter is released into that synapse, those neurotransmitters are then received by neighboring neurons, who are then either excited or inhibited by those incoming inputs. So the process is such that the signal has to be renewed at each new neuron. There's a new electrical signal has to be generated if that message is going to continue. And in order for that electrical signal to be generated, that receiving neuron has to receive sufficient excitatory input from its neighboring neurons. And neurons operate according to what we call the all or nothing principle. They either fire or they don't. So if a neuron does not receive sufficient excitatory input, then it's just not going to fire. It's not going to fire a weaker signal. It's just not going to fire at all. So it's the all or nothing principle. So in a kind of interesting way, neurons operate in somewhat of a similar way to a computer processor or a computer chip. It's just on or off, ones and zeros. It's a binary sort of operation, which is interesting to know about the brain. So at some level, at least, there is some similarity between the way the brain operates and the way a computer operates, although the parallels arguably start to break down at a certain point. But there have been a lot of researchers over the years who have compared the human brain to a kind of computer made of meat. They say the human brain is a computer made of meat. And some folks like that analogy, other folks aren't too keen on it. It has its defenders and detractors, but ultimately neurons operate in an on or off fashion. So just to recap the process, you have the dendrite, which is the structure on the neuron which receives incoming inputs from other neurons. And those inputs come in the form of neurotransmitters. And these neurotransmitters travel across the synapse. Neurons don't actually touch each other. And they travel across the synapse, and they cause that neuron to either be excited or inhibited by that incoming input. And the more excitatory input that is received on those receptor sites, then the higher the likelihood that that neuron is going to fire an action potential. The more inhibitory input that is received, then the lower the likelihood that that particular neuron is going to fire. So neurotransmitters tend to have either an excitatory or an inhibitory effect on the neurons that are receiving them. Here we see a zoomed in view of the receptor sites. We see a zoomed in view of the receptors, of one receptor site. So on the top, you see an axon and terminal button. So you see the terminal button there releasing a neurotransmitter, whatever it is. We'll say maybe it's serotonin or dopamine, whatever. We'll say serotonin. So here we have some serotonin molecules being released by the terminal button, and the receiving neuron has these receptor sites on its dendrites. So you have the terminal button releasing its neurotransmitter, and then the receptor sites on the neighboring dendrites of the neighboring neuron receive those incoming neurotransmitter molecules into these receptor sites that are kind of like a lock and key mechanism like you see here. So in order for those serotonin molecules to be received and in order for those serotonin molecules to affect the activity of the receiving neuron, those receptor sites of the receiving neuron have to be serotonin receptors. You're not going to fit a serotonin molecule into a dopamine receptor site, for example. So it's like a lock and key, and they have to match. So you have specially shaped receptor sites for each neurotransmitter. And we've identified, I think, over 60, if I'm not mistaken, around 60 or so neurotransmitters have been identified by science up to this point. And we see here in this list some of the more well-known ones, acetylcholine, for example, which is highly involved in attention and memory processes. Epinephrine and norepinephrine work together to promote fight or flight responses and arousal. They have an excitatory effect. Serotonin, highly involved in regulating mood, sleep cycles, and dreaming, by the way. Serotonin is an interesting neurotransmitter because it's involved in a lot of processes. A lot of folks think of antidepressants, and for good reason, because most antidepressants, not all, but most, do what they do by increasing the levels of serotonin at the synapse between two neurons. They increase free-floating serotonin between the neurons in your brain, and having more serotonin present there in the synapse, for whatever reason, for many folks, seems to be therapeutic in treating depression. So ultimately, the effect of most traditional antidepressants at the neurological level is to increase serotonin levels in the brain, or at the synapse more specifically. And you have your traditional antidepressants like Prozac, for example, and Zoloft, and many others, which are called selective serotonin reuptake inhibitors. And that's a mouthful, but what an SSRI basically does is increase the level of serotonin at the synapse. And there are some exceptions to that. There are some other antidepressants out there that we call atypical antidepressants, which do not affect serotonin, but the vast number of them do. I think the only atypical antidepressants that I can think of off the top of my head currently would be Wellbutrin, which increases dopamine levels in the brain, and also the antidepressant Effexor, which I believe affects both serotonin and norepinephrine, which is an interesting one. So just about all the major antidepressants affect serotonin with few exceptions. Dopamine is your neurotransmitter that is heavily involved in feelings of reward and pleasure. It's often thought of as the motivation neurotransmitter, is often sometimes thought of as the addiction neurotransmitter, because addictive substances and behaviors have a tendency to cause massive dopamine release in the brain. And anytime you see addiction processes, there's most likely going to be a connection to dopamine in that addiction. In almost all cases, anywhere you see addiction, you see dopamine, and addictive substances tend to cause massive dopamine release in the brain. Any sort of pleasurable activity, whether it's a substance like heroin or cocaine, or nicotine, or a behavior like sexual behavior, or eating, anything that feels good, really. Anything that feels pleasurable is connected to dopamine release. And that's the way a lot of recreational drugs work, is by hijacking the dopamine system, more or less. I mean, it's more complicated than that, but essentially dopamine is highly associated with addiction. So dopamine is kind of a double-edged sword, because it's a neurotransmitter that helps to keep us motivated towards pursuing our goals, but it's a system that can be hijacked and be related to addiction. GABA, which I usually just say GABA, I don't attempt to pronounce the full name of the neurotransmitter there, but I believe it's gamma amino butyric acid, so we just say GABA. But this is one of the principal, not the only, but one of the principal neurotransmitters involved in feelings of sleepiness. It's a neurotransmitter that is released in massive amounts. For example, if you take a sedative or a depressant like alcohol, anytime there's drowsiness or sleepiness, there's a high likelihood that GABA is involved in that process. Glutamate is an excitatory neurotransmitter that's associated with memory, as is acetylcholine, which I mentioned earlier, and it's an excitatory neurotransmitter. And endorphins, we have the natural painkillers of the brain, which are the system in the brain that's hijacked by drugs like heroin, your opioid drugs like heroin and fentanyl and morphine, any of those drugs that are derived from the opium poppy seed. We would refer to as opioids. And opioid drugs basically hijack your endorphin system and mimic the effects of your own endogenous morphine, which we refer to basically as endorphins. So endorphins are basically endogenous morphine. It's the natural morphines that your body produces on its own. And when we're in pain, you get an endorphin release. And this is part of runner's high, actually. Running can involve some degree of physical pain, right? Any sort of athletic exertion. And one thing that you'll often notice is that there's an endorphin release associated with activities like running. And you hear about runner's high, which is connected to endorphin release. Also, to some degree, dopamine as well. But endorphins are the natural painkillers of your body. So like I said, your recreational drugs that affect consciousness do so through affecting the receptor sites in your brain. And there are basically two types of ways this can happen. A drug can work as an agonist or as an antagonist. So an agonist would be a drug that enhances or mimics the activity of a neurotransmitter. For example, alcohol would be considered a GABA agonist because it enhances and increases the activity of GABA in the brain. On the other hand, drugs that inhibit the activity of a neurotransmitter, these are what are known as antagonists. Probably the most popular antagonist drug that comes to my mind would be caffeine, which blocks the neurotransmitter adenosine from working. And adenosine is an inhibitory neurotransmitter similar to GABA, which helps you to fall asleep. There are several inhibitory neurotransmitters, and adenosine is one of them that's involved in sleepiness. So the drug caffeine gets into those adenosine receptor sites and blocks them and keeps adenosine from being able to inhibit the activity in your brain. So essentially what's happening is caffeine inhibits an inhibitor. It blocks that inhibitory neurotransmitter, adenosine, from working. And just about all psychoactive drugs have their psychoactive effect through either agonistic activity at receptor sites, antagonistic activity. In some cases, you might see a combination of both. Another example, your opioid drugs, like I said a moment ago, they mimic and enhance the activity of your endorphins. So when you get something like heroin, for example, or fentanyl that gets into those endorphin receptor sites, it mimics the shape of those endorphins and it fools those receptor sites into letting it in and activating them. So that is what we would call an endorphin receptor site agonist is what heroin or any opioid drug would be. All right, at this point, I feel like I've been talking for a little while, and it might be a good idea to break this lecture also down into 2 videos like I did for chapter 1. So we've talked about the basic layout of the nervous system, and we've talked about the structure and function of the neuron. I know the structure and function of the neuron can be a bit confusing for folks, so if you have any questions about this, feel free to email me, or we can have a Zoom conference if you feel that it would be necessary to help your understanding. But there's still quite a lot left to cover in this chapter with regard to the layout of the brain and the various aspects of brain function pertaining to localization. For example, what parts of the brain do what? You have different parts of the brain and they have different responsibilities. So that's what we're going to kind of look at a little bit more in the part 2 lecture of chapter 2. So I think that'll be enough at this point, and I'll be working on our next video, which will zoom out from the neuron, and we will look at the brain at a more macro level. Until then,